Today's topic is about lipoprotein. First of all, our main aim is to solubilize lipid in hydrophilic medium because, as you know, our bloodstream containing the plasma is kind of hydrophilic medium and lipid couldn't be transmitted freely inside the bloodstream. For that reason, we need a kind of coat for that lipid in order to be transmitted inside that hydrophilic medium. How this transmission will occur? It's all mediated by the presence of lipoprotein. These lipids will be surrounded by hydrophilic surface and hydrophobic from inside, and in between there is a amphithetic, which is equal for hydrophobic and hydrophilic medium, thus forming a structure named lipoprotein. The lipoprotein way of transmission is from the intestine to the liver to the peripheral tissues. So intestine, the source from our diet consumption; liver, containing the stores and the synthesis of new lipoproteins; and the peripheral is a place of receiving the components of lipoproteins, which is either cholesterol or triglyceride. Great. Let's start by the first type of lipoproteins, which is the chylomicrons. Chylomicrons is the largest in size among classes of lipoprotein. This chylomicron contains in its surface ApoE protein and ApoC2, also containing a protein named B48. So this B48 is present only in chylomicrons among all the classes of lipoprotein. The chylomicron contains high concentration of triglyceride, literally high concentrations, and a small amount of cholesterol ester, which is an esterified cholesterol, not a free one. The ApoE receptor that's present for its binding of this chylomicron on liver receptors that will lead to the uptake of chylomicron by the liver in order for its synthesis and storing inside the liver. However, the ApoC2 is responsible for the binding of chylomicron on receptors present on the peripheral tissues that are responsible for delivering triglyceride into peripheral tissues from the stores or from our diet consumption. Also, this B48 is really important, where B48 will be responsible for the binding of chylomicron on surface of hepatocyte by another receptor. Second class of lipoproteins is the VLDLs. VLDL, very low density lipoprotein. This VLDL contains high concentrations of triglyceride, a little less than that present in chylomicrons. On the surface there is ApoE and ApoC2, same as chylomicrons, except for the protein present inside the B100 instead of B48. Also, the B100 responsible for the binding of this VLDL on receptors different than the receptors that ApoE responsible for binding, where ApoE binds the VLDL on LDL receptors, or also known as BE receptor, but B100 binds on receptors on the liver called HTGL. This HTGL will lead to the conversion of VLDL remnants into LDL molecules that we will talk about in moments. Also, this VLDL, as we mentioned, contain high concentration of triglyceride and a small amount of esterified cholesterol. Third class is the LDL, low density lipoprotein. This molecule contain high concentration of esterified cholesterol and small amount of triglyceride. This LDL previously, or in its nascent stage, contains ApoE and ApoC2, but it loses them upon its transformation, finally ending with only B100 protein that's responsible for its binding on liver beta receptors, in order to be uptaked by the liver. Finally, the HDL class of lipoproteins, the high density lipoprotein. This kind of lipoprotein is the healthiest one, and LDL is the bad one. It is the HDL contains high concentration of esterified cholesterol and low amount of triglyceride. The HDL, also known as the good cholesterol, and LDL is known as the bad cholesterol. Why that? Because the HDL is responsible for taking cholesterol from the peripheral tissues and storing it inside the liver. However, the LDL is responsible for taking cholesterol stores present inside the liver into the peripheral tissues. And as you know, as cholesterol was released into the peripheral tissues, it will pack up in the bloodstream and leading to the clots and narrowing of bloodstream. As you know, cholesterol in the blood will also block, for example, heart veins if it's present in excess. So for that reason, we called the LDL the bad cholesterol. This HDL, nascent form is named pre-beta HDL. It contains ApoE, ApoA1 and ApoA2. ApoE responsible for the binding of this HDL remnants, which means when the HDL take what it needs or when it performs its role and give, for example, the esterified cholesterol into the liver. This HDL will be empty from inside, kind of. This empty HDL is called HDL remnant. This HDL remnant should bind on liver receptors in order to be uptaken by the liver and resensitized again, and production of a new HDL through it. This binding will be mediated by the ApoE protein. However, the ApoA1 and ApoA2 role we will talk about in the upcoming videos. By this, we will end the topic of today. Hope I made it clear for you. Don't forget to subscribe, and I will leave links in the description for other YouTube videos. Thanks a lot.